Today, we're going to analyze the photosynthesis lab. We're going to try to answer the question, what happens to plants when the lights go out? What kind of reactions take over? So let's first of all take a look at some general information. First of all, you were supposed to make a prediction. What happens when light goes on plants? What's going to happen to the CO2 level? What we're really doing in this lab is trying to measure the amount of CO2 when a plant is exposed to dark and when it's exposed to light. So what happens to those levels? So what we all did is we made predictions, and then of course we're going to take a look at really what happened. So let's move on. Now if you can see, these are the average overall graphs that we did in lab. The one on the top one is when what happened to the CO2 level when plants were exposed to light, and the bottom one that's in blue, that's what is the line that generally characterizes what happened to the CO2 level when plants were exposed to darkness. So what did you think happened? And these are just representatives of an average of the entire class. So when you submitted them, we put them in Google Docs, and then what we did is we generated these graphs from all of your data. So let's look at the first one. Is this exposed to light or it's exposed to dark? And why would you say so? Well, most of you got this one pretty accurate. You pretty much said that when exposed to light, CO2 levels would actually drop because the CO2 is used up in the photosynthetic equation. So when we take a look at this graph in particular, what you're going to see is at first when the light hit, it kind of spiked, and then what happened over time is it slowly went down. And as it slowly went down, we found out, of course, that the CO2 level in plants, when exposed to light, actually went down. And the reason for that, many of you said, is because the CO2 is used to make sugars like C6H12O6. So that's what a lot of you predicted. So let's look at the other one. Now this next one, if we took a look at the CO2 of this line, this is what happens when we covered our apparatus, and then we collected over a 10-minute period what would happen to the CO2 level. Both are treated basically the same way. We had some aquatic plants. We put them in a beaker, surrounded them in water so we can keep the temperature constant. Shown a light on one, and then the other one, of course, what happened is after that 10-minute period, would wait 3 minutes, then would go for 10 minutes of showing light on it or exposing it to darkness, and then take those levels. So each one represents a 10-minute run, either in darkness or in light. And this is the one that many of you did not predict. This is when it exposed to light, and actually what happened is the CO2 levels went up. Many of you predicted the CO2 levels would stay the same. So that's really what we're going to be trying to do today, is try to explain what happened here in this graph when CO2 levels went up, even though it was in dark. Now let's take a look at our setup. We used, of course, the Pasco Spark data logger. We also used this contraption, our apparatus, where basically we had a moat of water around the beaker, and of course we had this stopper at the top, and of course the sensor. We measured just CO2 today, and of course right here this cable is attached to the data logger, and you can see one of the experiments that was going on. So that's how we treated this. This one happened to be in the light, as you can see. When it was in darkness, it was covered. So let's take a look at some of the results. This was our setup in light. What happened was we first of all exposed it to darkness and let it set overnight. We removed the cover for 3 minutes with the light on, so kind of get accustomed to it. And right in here is the aquatic plants. We put aquatic plants in this inside of the beaker. You can see it right down here. And it's filled with a little bit of water. And then this CO2 sensor, it has a hole in the stopper, and then we just put the sensor just above, and I don't know if you can see it or not, but the probe kind of comes right down to here, just above the water level. So this is the light treatment right here. And this water surrounding it acted as a heat sink, so we didn't fry the plants on the inside. And then, of course, in the dark cycle, what we came up with is the same kind of scenario. But in this case, we left them in darkness overnight. We went ahead and covered them so they were in complete darkness as possible. We took them out. We didn't actually uncover them. We just left them in there and the ambient light in the classroom. But it was covered. It's still in dark. We let it run for 10 minutes, and then we collected our data. And, of course, this is the dark treatment. And what happened? What does the data say happened? Well, let's take a look at it. Let's zoom in on it just a bit and a little bit more. There we go. This is the light treatment, and in this case, you could see that right here, it was 3,080 parts per million of CO2. And over here, we had 2,848 parts per million. And if you subtract those, it actually dropped, the CO2 level under light dropped 232 parts per million. And that's a fairly significant drop in CO2. But again, what we looked at today is if we zoom back out here, let's zoom back out. If we take a look at our equation, really we have what we call reactants over here. These are the reactants, and these are the products. So really what we were doing is measuring one of the reactants. This is what we are measuring. Most of you predicted the fact that as CO2 goes through this reaction, it needs this carbon from CO2, and that CO2 is used up in production of this glucose right here. So the C6 came from, of course, the carbon dioxide. So as the equation is running in this direction, CO2 that was trapped in the container, in the beaker with the stopper on it, was being used up. And in the meantime, of course, oxygen is given off. So when we look at this right here, it makes perfect sense because CO2 is being used up as it's making sugar. It's going to continue to drop. So that wasn't too much a surprise, and many of you got without much of a problem. So here's the one that was kind of stifling for a lot of you. And I'm going to get a little bit closer on here. Now, if we take a look at this one, this really wasn't very intuitive, because when we look at our reaction here, it's going in this direction. If it's in a dark environment, in other words, light's not being present, this particular reaction doesn't happen at all. This should stay constant. Maybe it drop a little bit, but to actually have it go up quite a bit, that was something that wasn't really expected. So in this case, when we look at the maximum level, or the final level, was 1,098 parts per million. And then over here, the initial was 656. You get an increase of 442 parts per million. So something else was really going on. Well, if you take a look at this equation, you'll notice that it has air that goes in both directions. Now, we know that the reason that we have the plants get the CO2 is animals are doing cellular respiration and are actually contributing to CO2. Here, the plants are making glucose, and also they're making oxygen. They're giving off the oxygen. That's kind of a byproduct. But and the only reason that happens is because we have this UV radiation coming in and is the energy supply to this in the chloroplasts. But when it's in dark, this shuts down. So what could possibly go on to be able to increase the CO2? Look at the equation. We'll notice here in this equation, if it goes in this direction, then CO2 is actually given off as a waste gas. So what are the plants really doing besides photosynthesis? And this is what came to a surprise to many students, is the fact that, wait a minute, if the lights go off, it can't go in this direction. The only way to explain it is they're doing respiration, cellular respiration, because they need the energy to carry on all the functions that goes on in the plants, whether it be growth, whether it is putting out seed, whether it's putting out roots, it needs tissues, so it has to use up this part right here, the sugar and the ATP, to be able to continue its life processes. So the reason why that this is going up is because the plants are doing cellular respiration. So plants in the dark, well, not necessarily in the dark, but don't need to, but when the light is completely shut off, it's going to start converting all of that sugar that is stored for itself. They're going to start using it up and generating CO2 as the byproduct, and of course provide the energy they need to be able to make the things they need to make in their life processes. So with that in mind, that's our mystery. What plants do when they don't have light, they start respiring. Now we can't do photosynthesis, or animals animals can't, but plants are able to do two things. They can do cellular respiration, and they can also do photosynthesis. So I hope that helps explain our mystery, and I hope it also explains why we did the lab that we did. And we'll see you soon. Thanks.